Hello everyone and welcome to The Wild Side. This is your host, Dr. Kight, and that was She and Him singing This Is Not a Test. And today I'm going to be talking about a variety of different tests in the context of wildlife forensics. And this is a topic that I've been increasingly interested in for a while now. I've always loved kind of the idea of forensics in general. I am a CSI fan, it's true. But also I am really interested in the whole wildlife forensics thing because this is the application of forensics to a variety of different things that has to do with, obviously, wildlife. So whether that is trying to catch people who've been poaching, or cutting down trees in a forest, or trying to track the movement of animals across different landscapes and they normally would be found in, trying to figure out if certain species have been mistreated in captivity, all sorts of things. So you can apply these tests in kind of a law setting, but then you can also use them to maybe figure out how to make some conservation and management decisions, how to take care of animals in captivity. There are lots of applications for these things. And one brief warning before I embark on today's show is that I've just discovered that my printer cut off the last bit of every single page in my little section of notes here. So that means that I might have some odd non sequiturs today, hopefully not. Just hopefully I'll remember everything I need to say, but sorry if I do say anything confusing. I blame it all on the technology. So to start off with, I want to talk about a very recent study that has just come out looking at a variety of tests that can be used in order to prove that animals that have been collected from suspected poachers really have been poached, or whether they're alive or dead animals, but to prove that these animals really have been gotten in an illegal way and not that they are pets or have been bought through a legal market. So really it's making sure that these animals are actually wild animals and not animals that have been in captivity for a long time, raised in captivity, sold through regular pet market sort of situations. And the interest of the authors in this topic was to try to figure out what kind of measurements can be used in order to distinguish between wild and captured animals, and also what kind of analyses need to be performed in order to link those characteristics with each other. So you might have one really good test, but then maybe you can't measure that in a particular animal for some reason, so you might want to have another test, or you might want to see how different tests kind of link up so that you can use multiple things all at once to really solidify your argument. And so that's what they wanted to do, is kind of collect all these different pieces of data and put them together in one big kind of meta-analysis. So this particular study was performed in South Africa, and obviously agencies in Africa but also around the world are really interested in the illegal trade of endangered species. It's a huge problem, particularly on that continent, but it does have effects elsewhere as well. As I'll talk about later, it is a growing problem in Southeast Asia also. And one of the big problems with endangered species trade and black market movement of animals is that it tends to be linked with other crimes also. So it's not just the animals themselves that suffer, it's not the habitats that suffer, but it tends to be linked with and bring in money for things like drugs and gun trade and terrorism and all sorts of other things that we don't want to have out in these communities. So it is a really important problem to kind of solve so that you can have these knock-on beneficial effects in other ways also. And we can use a lot of traditional crime-solving techniques to track down criminals after the fact, but this can offer many challenges and doesn't necessarily help us actually deal with the animals themselves. And one of the big problems with these confiscated samples is that the researchers often have to work with things that are diseased or dried out or quite sick, and that's because people who poach animals, whether they're killing them for meat or trying to collect them for the pet trade, they tend not to take very good care of them, and so the quality of these organisms declines, and that can actually make it very difficult for researchers to then go in and analyze them because they're not able to get very good samples from these individuals. And that's especially true if we're talking about a carcass that has been found somewhere, maybe contaminated with the blood of another carcass. Also, some populations might be completely wiped out by really intensive poaching efforts, and then you have very little data to fall back on because you might want to go to that original place where these animals were taken from and get some samples from relatives in order to find things out, but then there's actually nothing there for you to sample anymore. And another big problem is that unhealthy animals can then be released. So once they've been captured and held for a while in captivity, they don't do so well. They're declining in health. They might be with other animals and pick up diseases. And then once those are dumped back out into the wild, maybe because the criminals are trying to hide, maybe because they're giving them to people who are buying them, this can spread disease. So it can be a really serious problem. And that can also make it difficult for researchers to trace the origins of these things, because all of these sorts of patterns start to kind of intermingle and become quite confusing, because disease spread can be quite a complicated thing, and it's not always easy to tell, where did these things originate. So the basic idea is that we needed to establish a baseline information for what a healthy individual would look like, a healthy animal, and then be able to kind of use that information to then compare these captured animals from suspected poachers and say, right, does this animal look like that kind of healthy wildlife that we would expect if in fact it were recently captured, it were a legal thing, or does it look actually pretty bad because it has been captured and moved around and held in captivity for quite a long time. So you can have a big distinction between what an animal should look like and what it does look like after the poachers have gotten to it. And some of the things that you might think about when you're looking at this are things like, what are the animal's ranges? So should they be in the area where they've been found? What's their level of health? Are they quite healthy? And if they're not, what are the specific problems? And then, like I said, what do these things look like relative to animals that are in the wild and other animals that are known to have been imported and confiscated? So are they more like their wild counterparts, or are they more like the ones that haven't been doing so well because they've been in custody? So the particular study that I'm interested in is a study on tortoises, and there are 54 of them actually that were studied here. And these things were confiscated from poachers who were caught going from South Africa to Mozambique. And the poachers claimed that they were just out wandering around in the wilderness and they stumbled across all these animals, and so they went ahead and just collected them as they went. And that was just a couple of days ago, so they've only had them in captivity for a couple of days. They weren't out hunting at all. It was just that, they were there, they were taking opportunity to collect them when they saw them. And it was totally legal, and they didn't realize that nothing, that this wasn't allowed. Well, when the researchers found out about this, they said there were lots of things that kind of tipped them off that this was not actually legitimate activity and that these claims were probably incorrect. So one thing was that the animals were clearly non-native, so the poachers claimed to have found them in an area where they didn't actually live, and they also, one of them was very rare and quite hard to find, and so you wouldn't expect that people could just stumble across them by accident, because even people who go out in search of these things have difficulty locating them. Also, there were 3 different species across all these 54 tortoises, and it's very rare to find them all living in the same place, so it would be quite unusual for someone, again, to just be wandering through the wilderness and stumble across all 3 of these species. And also, to boot, there had been a recent turtle survey in this same area, and the researchers hadn't found much evidence of these species, and yet here were these novices saying that they were out there and they had no problem locating 54 different individuals. So this raised some serious suspicions about the origins of these turtles, and the researchers here wanted to collect a bunch of different data to see what things could they use to actually tell apart these individuals from their wild cousins, if you like. And the idea of this was that this information could be used in this particular case to hopefully convict the poachers, but also then could be given to zoo staff who might need to take care of these animals once they had been collected from the poachers, and it could be more generally applied to other situations that are kind of similar to this. So in the current study, what they did was they weighed and they measured and they bled the animals, and the idea here was to look at carapace length, so to give you an overall kind of body size of these individuals. And then for the blood measurements, they were looking at hematocrit levels and also hemoglobin concentrations. And these 2 things look at how many red blood cells there are and also how many proteins are available to carry oxygen. So basically this is thinking about whether these animals have been nourished enough, are they becoming anemic, are they in very good condition, or are they kind of puny, if you like. And one of the amazing things that they found was when they were actually in the midst of taking these measurements, they provided the turtles some water. And the turtles were so desperate for water, they were so thirsty, that they were drinking and they were submerging themselves even right in front of these people that were doing all the tests. And you know that normally turtles can be quite shy and they'll withdraw into their shells because they're really worried about potential predators. But these guys were so desperate for water that they were just staying out of their shells and doing this right in front of these people. So that was another big sign that these things were not in very good shape, and they probably had not been in very good conditions for quite a long time. So all these measurements that the researchers did on the 54 confiscated turtles, they also did on a subset of individuals that had been out in the wild. So they had the wild, free-ranging, supposedly fairly healthy individuals, and then they also had these ones that potentially had been in captivity all of this time. And they were able to then kind of create this composite index for individuals. So they looked at the average values for each one of these measurements, and then they looked at how much each animal varied from that average. So if you've got an animal that's varying in a positive way, then it's probably quite healthy. If you've got ones that are varying in a negative way, then they're probably in quite poor shape. And then on top of that, they used another method of analysis in order to look at how well all of these different indices could distinguish between the wild and the caught individuals. So if you kind of look at each of those subsets separately, and you kind of can see what are the types of averages, what are the types of values that we get for each of those measurements, for each of those subgroups, can we then just look at any random set of measurements and pretty accurately be able to identify that as a wild animal or a caught animal? And if you can, then that means you can potentially use these metrics later on other individuals that are captured further on down the line. So what they found when they were analyzing all their data was that the confiscated turtles, and in particular the females, had poorer body condition than the wild counterparts. And sometimes this was not all true for the males, but it tended to be more true for the females, and in general it was true of the confiscated turtles. And they also found that the wild animals had higher hematology values, which means that in general they were a bit healthier. So the confiscated animals were anemic and they had poor hemoglobin levels, so this probably suggests that they weren't getting the kind of nutrition that they needed. And the composite values varied, and the reason for this, seemingly, was that species identity and sex had a big influence. And so this is something that they obviously will have to take into account in the future, with that you can't just look across all tortoises and kind of have a general kind of scheme for measuring things. You have to look at the females and males separately, you have to look at the different species separately, because each of those will have kind of a tendency to have certain measurements, whether it's body size or weight or the hemoglobin and hematocrit levels. And they did find actually though that they had a pretty good correlation between the values and the animals in the species that they had the biggest sample size in, which is encouraging because this suggests that if they go out and are able to get even more of these measurements and kind of refine their technique a bit, then maybe they can come up with metrics that are even more specific and accurate in the future. And the discrimination thing that I was talking about, so figuring out whether this is something that can be used to actually distinguish wild from confiscated, this also varied quite a lot, so it wasn't always as reliable as they might like, but in general it was pretty good. So what you could do was not only include those measurements that they took, but also kind of control for the sex of the individual and the species of the individual, and that allowed them to then have pretty good classifications of whether animals had been wild or whether they had been confiscated. And so this means that yes, you can kind of look at a turtle and take some measurements of it and say with reasonable certainty whether or not this animal has just been taken from out in the wilderness or whether maybe it's been held in captivity for a while by potential poachers. So this was quite a nice thing to show, because all of these turtles that were confiscated did not have noticeable signs of trauma. So if the poachers had a pretty good argument in court and they presented these turtles that didn't have any outward signs of trauma and they didn't have any other evidence to back up the claim that these were not in fact taken from where the poachers thought they were taken, then it might be very difficult to actually convict these guys in a court of law. But because they had all this forensic information, they could in fact say, you know, despite the fact that these turtles don't look like they've been traumatized, they don't look like they've been injured, they are in really poor shape, they aren't as heavy as they should be for their body size, they don't have the right blood values because they've been mistreated, and this was very useful for showing that these guys had been held for quite a long time by these potential poachers. And another nice thing about this is that all of these things that they did were very quick and very portable. They're also well-developed and reliable methods, and they can be trusted. And this means that potentially a large number of people could go about the countryside and do these sorts of things wherever and whenever necessary. And this is really encouraging because it means it can be rolled out as a technique not just in South Africa, but you know, wherever else someone might need to do this sort of thing. You can just have a little satchel full of the tools you need and dispatch them out to the field and let people take these measurements as poachers potentially are caught. And another good thing was that the concepts associated with these tests could be explained to people in court. So the authors felt, you know, that obviously you don't want to have something that's really, really complex and confuse people who don't understand what these tests are and what they mean. And the fact that these are relatively simple, you know, measuring a carapace length, that's quite a nice thing because you can easily explain to someone that this is a legitimate thing and it's quite an easy thing and it's pretty straightforward. And even better is the fact that all these different parameters that they took measurements of, the 4 different things, were supporting each other. And this is also going to be quite convincing in a court setting. And in fact, the actual outcome of this case was that the confiscated turtles were so completely different from the wild animals overall that it was very clear that they'd been in captivity and mistreated for quite a long time. The hematology results showed they had been desiccated and starved. And so this allowed the poachers to be convicted for quite a large fine, I think. They didn't have to go to jail, but they did have to pay one of the largest fines, if not the largest fine in the history of that area. So it was a successful court case, which sets a really nice precedent for using this sort of thing in the future. And the only drawback is that you just have to make sure that researchers are focusing on the right variables. They've collected sufficient data to really separate the wild-caught and the captive animals, and that they know how to interpret these sorts of data. But for sure, this is definitely a really good first step in using these sorts of analyses on turtles and potentially other animals as well. Welcome back to The Wild Side. That was Alana Davis with Turtle. And today on The Wild Side we are talking about wildlife forensics. And the next study that I want to talk about is one in which researchers have used a relatively new technique called DNA barcoding in order to identify an endangered species that people try to illegally sneak through customs and trade on black markets. And this whole technique was used on a plant, and actually a group of plants, that produce something called reserpine. And reserpine is an alkaloid that can lower high blood pressure. And it's particularly common in a plant called the Indian snake root. And what harvesters do is they find these plants and they take their roots. And they do it when the plants are about 3 to 4 years old. And that's because that's when they have an abundance of this alkaloid in the roots. And so they dig up the roots and chop them off. And obviously, once a plant doesn't have its roots, it doesn't last too long. And so this causes the plants to die off. So this is pretty unsustainable as far as harvesting goes. And as a result of that, snake root has been really over-harvested. So all the roots that have been taken have caused all these adult plants to die, and there aren't new ones coming in because they haven't had a chance to breed yet. And so actually this plant is just being completely eradicated in big patches of its habitat. So this is particularly common in Southeast Asia, although different species in this genus do grow around the world, but Southeast Asia is where there's a really big problem with this snake root harvesting. And in 1993, which is the most recent estimate of these sorts of figures, researchers thought that maybe 400 to 500 tons of the plant were being gathered in just a small portion of Southeast Asia, so just 4 or 5 countries out of the many that are there. This is obviously quite a big problem. That's a huge haul, and the plant is now considered endangered by IUCN, and it's also on the CITES list of untradeable species. And despite this black market kind of activity does continue. So the big problem is that people want to be able to tell when someone is treating the roots, and they want to be able to look at samples and say, yes, that is an Indian snake root, so this person should be arrested and this sample should be thrown out so that it doesn't keep stimulating the market. But it's really difficult because traditional identification requires that you look at things like flowers and fruits. But like I just said, snake root is treated as these sterile roots, and they're very hard to distinguish taxonomically. You can't just look at that and tell it apart from any other big root that it might be thrown in a bucket with. So this is where the latest molecular techniques kind of come into play, because if we can take samples of the DNA of these species and find particular markers that make them quite obvious, then that would be a really good way to actually make these identifications and not worry about the physical aspects of things. And as I said, there are other species in this genus from around the globe, and in fact there are about 73 different species in the same genus, and they grow in various forms all the way up from little herby-type plants to big, great big trees. So if they were to look at all of these different species and get kind of samples from around the globe of things that might kind of look like this particular species and be quite hard to tell apart from them, and if they also focus specifically on the species that are most commonly traded and most closely resemble this particular form of Indian snake root that is of interest, and they were to still be able to find something that distinguishes this one species from all of these others around the globe, then that's probably going to be a really, really useful thing for telling it apart. So obviously with other species you would expect, yes, the DNA would be quite different. But species that are in that same genus, species that are particularly closely related, those might be a bit more confusing. So if you narrow down your search and just look for whatever you can find that will distinguish between snake root and those other things, that should give you the result that you're after. And so that's what these authors did. They collected leaves and roots of these plants. Some of them were from actual samples that had been confiscated. Some of them were from herbarium specimens. Sorry, I can't speak all of a sudden. And these were, obviously prepared quite a long time ago, and chemicals were sprayed on them. They were put in the herbarium. So these are really hardcore samples. So if you can get something out of that, then you can probably get something out of whatever you confiscate from potential poachers. So they extracted DNA from that, and they used a procedure that allows them to amplify particular sections of the genome that are of interest to them. And so what they did was they particularly were focusing in on a portion of the genome that they know changes quite rapidly. And so you would expect then that it would only be similar among individuals in the same species than among individuals of other species, because it probably will have changed from one species to the next. And therefore be quite distinct and quite different. So they identified some sections that were like that, and they amplified them so they had lots of copies of this to work with, lots of material to analyze. And then they were able to compare all these things across the different species and kind of isolate from those subsections of DNA which ones are the best, which ones do we think will be the best for these sorts of analyses. And then they went on and actually tested those on some known samples from across the plant's entire range to see whether the test that they developed could accurately say, yes, that is the snake root. And so then they would know that they could actually use that in the future in this kind of wildlife forensics setting. So as I said, they were trying to isolate these particular DNA sequences, and they did find one in particular that had lots of variation. And variation is quite nice because it means that you have lots of things that you can test for. So it's pretty easy to make a mistake if you don't have a whole lot of variation, because that means that you could, very, very easily classify something as A when it's actually B, just because there are only two choices. And so if one little mistake happens and it looks a bit more like B than A, then you've accidentally classified it the wrong way. So you want to have something that has lots of variation and allows you to really be certain. So they found one of these, and then within that one little section of DNA, they further narrowed down a particular portion that had another section that was of interest and quite useful in doing the identification. So just to kind of give you the basics of DNA, I'm sure you know this, but just in case so we're all on the same page, DNA is made up of these base pairs, these 4 different base pairs, and so they found a stretch of several different base pairs, and that's that sequence of interest that they focused on that had all the variation. And then within that stretch, they further narrowed in and found a subsection of just a few base pairs that they could then really focus on. And one of those sections was a section where several base pairs were frequently deleted, so the DNA section would be a bit shorter than normal, and another they had several base pairs that were inserted, so that DNA section would be a bit longer than normal. So then that means if they are looking through each specimen and they take out that little fragment of DNA and they measure how long it is, they kind of know, generally speaking, how long it should be. And in this case, they found that the fragments should generally be 137 to 177 base pairs long overall, given the fact that you sometimes have the things that are deleted and you sometimes don't. But then in other species, they found that the range was quite different. So that means that, without a doubt, when those ranges are not overlapping with each other, and they weren't, the other ones were 146 to 160 and 156 to 176. So these are very distinct lengths. So you can look for the specific length of that species that you're interested in, that Indian snake root, and you can tell that it's not the length of those other species, and that's what allows you to make the identification. And so that means that you could reliably and consistently identify this species of interest. And then when they did this, they did find that it was really reliable, that they were able to say, yes, this is snake root to all of those known samples that they were testing, including also a sample that they thought was snake root. It had been confiscated in customs, and they did an analysis and proved that, yes, it actually was. And they also found that despite the fact that some of these samples had been treated with chemicals, this wasn't an issue, so the test was really robust and it was able to stand up to these sorts of additives that had been put on the samples after the fact. And they also found that they could do an analysis on both roots and leaves of the snake root plant, so it didn't even matter what portion of the plant they had a sample of, they could still do the test. And one other thing that was quite interesting was that there is a previous paper looking at Indian snake root and saying, we actually do think that we can look at some bits of the morphology of the way the veins are and the way that the microstructures are in the leaves and in the roots. So, obviously that's going to be quite a difficult thing to do. You might not have a big enough sample to do that on, but in places where you do have that, then that means you have two tests that you could use in conjunction with each other on this one particular species, which will be really useful if they do confiscate this in large amounts. So overall, this was quite an encouraging study. They were able to do these tests very successfully on a range of different samples, and they were able to really clearly distinguish snake root from things that look like it and are also closely related to it. And researchers in the future, they say that what they would like to do is add even more species to their database to make sure that there aren't other specimens out there that do look like the snake root in terms of the DNA. So more of these other species in the same genus as the snake root, even other species as well, because you never know, maybe there is some unrelated plant that for some reason does have this same genetic feature. So the more plants they add to this database, the better. And that's actually something you hear quite a lot of whenever you're looking at these kind of DNA barcoding type studies, is that people say, look, this is a great technique, but it really depends on having lots of data available in the database so that we can compare these things with as many other things as possible. So in the future, that's hopefully what researchers can do. Forensics people can go ahead and use this test for now, and they can probably have very high validity and accuracy, but in the background, if we keep adding these other species, then the test will only get stronger and stronger as they go. And that was Audra Mae singing Snakebite, and you are listening to The Wild Side, and today we are talking about wildlife forensics. And the next study that I would like to talk about is one in which researchers were looking at animal tissues and were aging them using isotope techniques. And what's really interesting about this is that the isotope techniques relied on certain signatures that were created by anthropogenic activities, and in particular anthropogenic nuclear activities. And this can be interesting and useful in a variety of ways, and one of them is, again, because I'm talking about wildlife forensics, obviously, you might guess it's quite important in looking at poaching and samples from animals that potentially have been poached and trying to figure out where they've come from and where these poachers are operating. So this study, as I said, it uses isotope techniques, and the whole reason the isotope techniques work is that they are based on the fact that you've got different forms of certain atoms out in the wild. So for carbon, which is what this study was looking at, most of the time you've got carbon with 6 neutrons and 6 protons, and that gives you a type of carbon known as C12. And C12 is the one that is in like 98% of all the carbony stuff that we can find on the planet. However, there is also another type, actually there are a couple of other types, and one of these is C14. And this is produced in the atmosphere when neutrons interact with N14. And N14 is already up in the air, and you've got neutrons that get released that are up there, and the neutrons bang into the nitrogen and they create this carbon-14. And this happens when the products are being exposed to lots of energy. So things like cosmic rays, also bombs, and nuclear reactors as well. So some of this will happen naturally, like the cosmic ray stuff. Other stuff is caused directly by human activity. And once the C14 is created, it's then incorporated into the tissues of living organisms because it functions just the same way that the C12 functions. So plants will take it up directly and it's not an issue. It still works just as well as the other stuff. And so plants will have it directly from the environment, and then other organisms will have it because they eat the plants, and they'll incorporate the plant carbon into their own bodies as well. So it does propagate up the food chain. Now, atmospheric variations in C14 have been measured for the last 60 years around the world because once people realized that these were affected by nuclear activities, then a lot of nuclear power plants were fitted with these devices that allowed researchers to measure the C14 in the atmosphere. And this allows them to create something called bomb curves. And bomb curves are just kind of plots that trace the abrupt rise and fall of carbon as a result of anthropogenic activity. And this activity can be anything from testing nuclear devices to dropping a bomb during war to doing any kind of regular nuclear activity that would happen on the planet, even things like whenever a nuclear reactor fails and there's a horrible event like what happened at Fukushima. So all of those ranges of things that we engage in cause these bomb curves to have these rises and falls. And you can also see these same things happening in organisms. So you've probably heard of carbon-14 dating, and this uses, it's based on that same stuff that I was just talking about, where you can just kind of look at the ratio of carbon-12 to carbon-14 within an organism and then tell about how old it is. Well, you can also look within particular organisms at variations from one part of a tissue to another, one part of a body part to another, and look at the change of carbon-14 over time. And this will allow you to see that same kind of variation, that rise and fall of carbon-14 as these things are incorporated into the body as the carbon-14 levels vary in the environment. So what the researchers here were interested in doing was looking at those bomb curves within organisms, but exploring some new types of tissues that hadn't previously been looked at, and also kind of figuring out whether they can maybe, what kind of scale of measurement can they make, how fine-grained could they get, how much data could they extract by looking at these changes over time, and what kind of information can you really extract from that, and if you do an analysis, what does it mean, what can you really tell about these animals? So what they found, well actually, sorry, I should start with their methods. What they did was that they collected these inorganic tissues and also organic tissues. So things like dentin and enamel associated with tooth structures, keratin, collagen, and also bulk plant tissue. And they subjected these all to chemical reactions. And the chemical reactions ultimately yielded carbon dioxide. And that was what they needed in order to test, because the carbon dioxide contains carbon, obviously, and that's what they're looking for, so they can isolate how much C14 is in that sample. So they took that carbon dioxide, they purified it, they turned it into graphite, and that was the form in which they could measure the C14. And then this allowed them to calculate the ratios of C14 to C12, so just like in regular carbon dating. And what they found was that from about 1955 onwards, the wildlife bomb curves are very similar to those that were created from the data that these sensors had detected, that these sensors had collected at these nuclear facilities. So there was quite a lot of agreement between the stuff that people were collecting from the air and the stuff that the animals were showing within their tissues. However, samples before this time weren't really accurate. So carbon-14 dating in general is fine, it's a great technique, but in terms of actually looking at these bomb curves, it's only from about 1955 onwards that you can get a good idea of kind of these anthropogenic influenced curves of carbon-14. Another thing that they noticed was that data from the different hemispheres sometimes was very similar, but also sometimes it was quite different. And the reason for this variation, and also the reason for variation within the types of samples they looked at, was that you have different bomb testing locations, you have different amounts and types of circulation of C14 in the atmosphere, and so these things can cause some flux in terms of the actual amount of carbon-14 that is available to the organisms when they actually go to take it up. So even though we know that, for example, a lot of nuclear activity happened in Japan when the nuclear reactor blew, that might not necessarily be where all of those things end up, because they get moved around in the atmosphere, and that the C14 could fall down elsewhere in the world, and that's where these curves would actually reflect the activity. So that's why there was the spatial variation. And they also found that there was some difference in terms of which samples had the best match between their curves and the curves shown in the data from the nuclear facilities. So grass samples were the best, and then soft tissues and collagen structures, and then apatite that had been taken from the dental bits, so things like teeth, obviously, and then also the keratin was kind of in that same range. Now, one thing that they kind of did that was interesting was they found some mismatches in previous samples. So, for example, they had this blue monkey that had been labeled as being collected in 1962, but based on the bomb curve data was obviously from before 1955. And this is kind of an interesting side effect, if you like, of this study, was that they found that actually the way that we label some of these things might be a bit misleading. So probably the museum had labeled the monkey in terms of its date of acquisition and not its actual age. And so people who were doing analyses, researchers who were using samples from herbariums and museums to do analyses on whatever, might actually have some incorrect data because of this labeling system. So it's quite a nice thing to know about, and potentially something that could be rectified by using these sorts of bomb curves and sampling those specimens a bit more closely. Another thing they looked at was that you could use multiple measurements from within the same organism, as I mentioned earlier, in order to look at tissue growth rates. And this is quite interesting because you can then kind of look at growth rate from one specimen to another specimen in the same species and see how much variation there is. So this was particularly good in things like elephant tusks, although they also were testing hippocanines and other various bits of elephants. And they found that in the elephant tusks in particular, there were these variations in growth rate just within the same species. And that is probably due to differences in conditions, so things like diet and stress levels, although it could also just be ontogeny, or just kind of variations in development. And the reason that they were particularly focused on the differences in conditions was that some of these animals one of the animals that they tested was a captive animal that had been associated, I think, with a zoo or a circus or something like that. And the other was one that had been out in the wild. And so you're probably now beginning to see why this is so interesting from a wildlife forensics perspective, because if there is this variation in growth rate, then you might be able to get an idea of whether animals were in captivity for quite a long time, whether they were in the wild, even maybe what part of the world they came from, what sorts of foods they were eating. And this could potentially allow you to track animals that had been captured, or to figure out where poached animals had originally come from. So just to broaden that out a little bit, it's not just relevant to wildlife forensics. There are actually just generally a lot of information that you can get from this stuff. So the researchers think that by looking at these variations in isotope profiles, they could look at changes in diet and also water use, and this would then, you could extrapolate that to figure out changes in precipitation and vegetation growth. So if you're thinking about people who are interested in really historical samples, they might be able to reconstruct the diet and ecology of fossil animals based on these tusks or bones that are left behind. So this could allow us to extract both figuratively and literally a lot more information from these fossils that are left behind by these animals. So we wouldn't just know what they look like, but also what they were doing and what type of landscape they were inhabiting. And you could also detect changes in food preferences or habitat use, and this could be something that's useful kind of in real time for thinking about conservation and management. So if you've suddenly got a population of animals that is utilizing a whole new part of their habitat than they ever were in before, this could be quite important for figuring out where to kind of decide a park should be labeled or where to keep people from entering so these animals can be left in a safe place. And finally, the thing that most of the press have been covering in terms of this story was that you can potentially use this technique to determine the age and also the origin of ivory. So you can look at these samples that are being sold illegally on the black market and figure out from the diet and the kind of climate and the growth rates where are these animals coming from. And that can allow you to then go back to those places on the ground and try to protect those locations, try to keep out poachers, and maybe you can also use similar methods for hair and horn of other species. So this wouldn't just be an elephant-specific thing, but could potentially also be quite useful for rhinos and other species that are put upon as well. So I think because I'm running out of time, I don't have time for 2 more songs, I don't have time to dwell as long as I want on this last study, so I'll just kind of quickly summarize it and then finish up with one last song. And the final study that I want to mention is one in which researchers used a phyloforensic approach to identify the origins of trade animals that may or may not be legal. And this is something that they did in Southeast Asia, where there's a huge problem with the illegal trade of wildlife. And these wildlife species are used for skins and meat and charms and medicine. They're also taken as pets. And some of this is legal and some of it is not legal. And one of the species that is particularly suffering from this is monitor lizards in the Philippines. And these guys are of conservation concern because they've been really heavily exploited. So what these researchers wanted to do was get samples from these guys and figure out where they actually came from and what species they were, and then to ask the people that were selling them where did they think the animals were from. And that was just to get an idea of, how aware are people of what things are being sold and where they're coming from, and how aware are the buyers of whether or not they're buying something that is endangered or something that's from really far away, something that's quite common, and to just get kind of an idea of how perception matches up with the reality. And what these guys did was they went into a marketplace and they took all of these samples and then did a brief interview with the people, and they made it clear that they weren't going to turn them in. They made it clear that they didn't want to buy anything. They just got the sample and then took off again, so hopefully it didn't influence their results any. And what they found was that there were 5 species of lizard that were being sold, and the most common were the ones that were the most widespread throughout the Philippines. So as you might expect, the ones that are quite easy to get your hands on are in fact the ones that were being sold the most. However, there were also some species that were identified from really far-flung locations, things like the Asian mainland and Borneo and Sumatra, and some of these were not native, obviously, to the Philippines. And actually one of them was very rare, and another was from an area that was nearly completely protected within a national park. So you can see how this technique would be quite useful, not even within this context of kind of matching up perceptions with realities, but also actually tracking down where are these animals being harmed or put in harm's way, so they can figure out where to protect them best. So what was interesting when they looked at the perceptions of the sellers was that only about half of the samples were actually from the places where the sellers said they were from. And some of this, they think, is probably because the sellers want to drive up the prices. So if they can pick a location or pick a species that suggests that the animals are rare and hard to come by or hard to access, then they can make buyers pay even more money. And what's quite problematic here is that the specimens had come from throughout the Philippine archipelago, which suggests that the hunters or the sellers or both were very resourceful and that they could probably get their hands on lots and lots of individuals and species, which is not very good news for these lizards. And there were obviously some species that couldn't be included in the study because there are lots of species, there are lots of lizards out there. And so like these other studies that I mentioned, the researchers say what they need to do is do lots of sampling, build up a really big database so they can do better comparisons and come up with a more specific kind of map of where these animals are coming from and where they're in harm's way and the most. And one of the things that would be quite interesting about doing this was that a bigger study and a continual study would allow them to go out to different markets as well. So this particular market they looked at was in Manila, but they could also visit other markets and see how much variation there is from one place to another, because maybe some markets are worse than others, maybe some are better than others, and they could kind of compare at that level as well and see all the different sorts of influences on the lizards that are being sold and where they're coming from. So I think that I will wrap up for today. So hopefully you know a bit more about wildlife forensics, the sorts of techniques that can be used and why they can be quite helpful. This is quite an exciting new field, I guess. I mean, it's not completely new because forensics have been around for a while, but more and more we're seeing that these things are being used in wildlife settings to help with conservation and management and reconstructing kind of the history of these fossils that are being discovered. And it's just really fascinating to see all the new uses that people are coming up with of these old tests, and also the new tests that are being developed to kind of address these particular problems within this field. So I will put all this information that I discussed today up on the website. This is the last you will hear of me for a couple of weeks because I'm going to be in the US, but I did already pre-record 2 shows to play while I'm gone, so hopefully if you are still interested in hearing about the wild side, then you can tune in next Wednesday and the Wednesday after and hear a bit more about the exciting stuff that I'm talking about in those shows. So with that, I will leave you with Aimee Mann singing Little Bonds.